DNA, or deoxyribonucleic acid, is a complex molecule that carries the genetic information of living organisms. The structure of DNA is known as a double helix, which was discovered by James Watson and Francis Crick in 1953. The DNA structure consists of two long strands that wind around a common axis, forming a double helix. Each strand is composed of repeating units called nucleotides. Each nucleotide consists of three main components: a 5-carbon sugar called deoxyribose, a phosphate group, and a nitrogenous base. The nitrogenous base can be one of four types: adenine, guanine, cytosine, or thymine, abbreviated as A, G, C, or T. In the DNA double helix, these bases pair up according to specific rules. Adenine pairs with thymine, and guanine pairs with cytosine. This double helix structure of DNA provides the molecular basis for storing and transmitting genetic information in living organisms. Multiple chemical bonds stabilize the DNA structure. In each DNA strand, nucleotides are connected by phosphodiester bonds. These bonds form between the phosphate group of one nucleotide and the sugar of the adjacent nucleotide. These bonds form the sugar-phosphate backbone of DNA. The two DNA strands are connected by hydrogen bonds between complementary bases. Adenine always pairs with thymine, forming two hydrogen bonds. And guanine always pairs with cytosine, forming three hydrogen bonds. This base pairing rule is called the complementary principle. The complementary principle is the basis for accurate DNA replication. Phosphodiester bonds and hydrogen bonds work together to stabilize the double-stranded structure of DNA. DNA, the blueprint of life, carries genetic instructions for growth, development, function, and reproduction in all living organisms. The functional units of DNA are genes, which encode the information needed to build proteins. Gene expression is the process by which the information in DNA is used to synthesize gene products. It consists of two main stages: transcription, where RNA is produced from DNA, and translation, where proteins are synthesized from RNA. DNA's unique characteristics, such as its ability to replicate precisely, enable the transfer of genetic information from one generation to the next. Variations in DNA sequences lead to genetic diversity, which is the foundation of evolution and adaptation of organisms to their environments. Today, understanding the structure and function of DNA has applications in various fields, such as medicine, agriculture, and biological research.